Hello everyone, this is the second video in our presentation on gluconeogenesis, glycogen synthesis and degradation and the pentose phosphate pathway, and we're going to talk today about glycogen synthesis and glycogen degradation and the regulation of those processes. So what is glycogen? Glycogen is a polymer of glucose. So glycogen is basically composed of linked glucose residues, and the linkages between the glucose residues are in one of two types. Either the alpha-1,4 type linkages, where the carbon 1 of a glucose is linked to the carbon 4 of the glucose adjacent to it. So we call that an alpha-1,4 linkage, or it can be at the branch points a alpha-1,6 linkage, so a carbon 1 being linked to a carbon 6. And glycogen is then the storage form of glucose in animals and humans, and it is analogous to starch in plants. So the way in which plants would store energy would be to produce starch, and similarly mammals would produce glycogen to store energy. Glycogen consists of granules of high molecular weight, and it is within these granules a very large amount of glucose residues can be stored to be quickly available as a source of energy. So glycogen is synthesized and stored in the liver and muscles. Glycogen in the liver is very important as a source of providing glucose when blood glucose levels become quite low, and of course glycogen in the muscle is very important when doing strenuous exercise. It allows it to be quickly mobilized and act as a source of energy. The synthesis and breakdown of glycogen occur in different paths, and they are catalyzed by completely different enzymes. So unlike with glycolysis and gluconeogenesis that we've spoken about, these are not amphibolic pathways because they occur by completely different paths, the making and the breaking of glycogen, if you will. So I like to think of the synthesis of glycogen as adding linkages to a chain, and glycogen is synthesized from glucose, as we've said, but glucose has to be in a kind of activated form to allow the transfer to a sugar chain by formation of sugar nucleotides. So glucose is phosphorylated first to form glucose 6-phosphate, and that of course is the first reaction of glycolysis by the hexokinase enzyme, and from there it is isomerized to glucose 1-phosphate. So that is phosphoglucomutase is the enzyme involved there. It doesn't require energy, and from there an enzyme called UDP glucose pyrophosphorylase catalyzes a phosphoanhydride exchange, and this is driven by the pyrophosphate hydrolysis. So what are we talking about? Well, in this case it is a UTP, is the trinucleotide that will yield the energy for activating glucose in order to allow it to be built into the growing glycogen chains later on. And what happens is that a glucose 1-phosphate produced from glucose 6-phosphate through phosphoglucomutase will receive this phosphoanhydride exchange, and from glucose 1-phosphate we will form UDP glucose, and you will see the two phosphates from the original UTP is released as a pyrophosphate, which is then split into two inorganic phosphates, and what we have at the end is a UDP and a glucose. So this will basically be the building blocks of the growing glycogen chains. We will see as the glycogen is synthesized, it is UDP glucose that comes in, the glucose is added to the chain, and the UDP will be released. So let's start now with where glycogen synthesis actually starts, and it's initiated by a protein called glycogenin. So what is special about this protein is that it is a self-glucosylating enzyme. So it is an enzyme that is produced, it can undergo autocatalysis, and it can self-glucosylate, and what that means is that it will start adding glucose residues to itself. So the first glucose residue is added to a tyrosine hydroxyl group, and that will allow a point for the glucose chain to start growing. So it can create short chains of glucose attached to itself, and when it does this, once the chain reaches more than 10 glucose residues, they will act as a kind of a primer for glycogen synthase to elongate them. So once glycogenin has 10 or more glucose residues attached to it, the glycogen synthase can attach to this complex, and it'll allow the addition of a lot more glucose residues to it. So the glycogen synthase will then transfer those glucosyl groups from the UDP glucose that we saw on the previous slide to the C4 hydroxyl at a non-reducing end of the growing glycogen strand. This is how more glucose residues are added to this growing chain. So how this works, UDP glucose binds to a branch end through the action of the glycogen synthase enzyme, and UDP is released, and the branch is now one glucose residue longer. So we have the UDP glucose we saw the synthesis of a couple of slides ago. We have our growing glycogen chain over here, and through the action of glycogen synthase, the UDP is released, and the new glycogen residue is added to the end of the chain, and our glycogen is now one residue longer. So finally, we will also need a chain branching enzyme. We said that the glycogen particle is a highly branched structure. So the elongation of glycogen can have branches. The branches help keep the polymer soluble and creates a lot of chain ends for rapid release when glucose is needed. So these are two important points to remember. The polymer is highly branched, that granule, and this is because it has to remain soluble. So even though we want a granule with a lot of glucose residues, which can become something with quite a high molecular weight, we still need it to be soluble inside the cell. And the high amount of chain ends is required, as we'll see a little bit later in the degradation. The most important enzyme for the degradation of the granule is an exo-acting enzyme, so it needs a chain end to attach to allow it to break down and release the glucose. So it is very important that it is highly branched, the granule. So the branching enzyme then forms branched glucose chains by transferring short sections of glucose of around 7 residues from one chain to another. The attachment of the secondary chain is at a position 6 of the receiving glucose, and this then forms a alpha-1,6 linkage. So what we're showing here is this was a growing chain of glycogen. It was all in the alpha-1,4 formation of the bonds, so all of the little bonds are alpha-1,4 bonds, but then the branching enzyme will cut at least 7 residues from this growing chain and attach it somewhere else. And this new bond that it forms here is onto a carbon 6, so that makes that then a alpha-1,6 bond that is being formed, and this is how branches are formed. So how then is the glycogen particle built up? So just briefly on the structure of the particle, it starts as this central glycogenin molecule, which is what we see here, and of course we said this autoglycosylates, so it starts forming its own glucose residue chain, and then the branching will start, or the synthesis will start by the glycogen synthase, and the branching will follow that. So starting at the central glycogenin molecule, glycogen chains, 12 to 14 residues, can extend in tiers. The inner chains have 2 branches each, so every inner chain will have 2 branches that you see over there. So this then would be the first tier, the second tier, the third tier, and what we illustrate actually here is that only 5 tiers are shown, but there can be 12 tiers in a mature glycogen particle, with the outer tier being the only one that is unbranched. And in this way, a glycogen particle can contain about 55,000 glucose residues and be approximately 21 nanometers in diameter. So it is quite a large granule inside the cell, and yet because of this very specific branched structure, it remains both soluble and it has a lot of chain ends. You'll see all of the little branches have a chain end to which the degradation enzymes can attach. So let's talk now about glycogen breakdown, and there are several enzymes involved here. The first one we're going to discuss is an amylase, an alpha amylase. So alpha amylase is an endoglycosidase. So what that means is that any endo enzyme will act within the chain somewhere. It doesn't act on a chain end, it acts within a polymer chain. So this is an endoglycosidase enzyme, and it cleaves dietary amylopectin, form of starch, or of course it cleaves glycogen to maltose, maltotriose, and other small oligosaccharides. So this would be a glucose residue. If we have a dimer of that, two glucose residues bound in a alpha 1,4 bond will make that a maltose. And once you have 3 residues or 4 residues, we refer to that as small oligosaccharides. So, as I said, this is an endo-acting enzyme. It will easily hydrolyze the alpha 1,4 bonds as long as they're not close to a branch point. When you get close to a branch point, its activity is reduced, and you actually need a debranching enzyme that will cleave those areas around the branch. So when amylase acts, it'll digest away everything that is not close to a branch, and that leaves you with these areas around the branch points, which we call limit dextrins. And we specifically need the debranching enzyme to remove those limit dextrins. Now, the most important enzyme in breaking down glycogen is called glycogen phosphorylase. It is a phosphorylasis reaction and not a hydrolysis reaction. So what we've spoken about quite often is enzymes that use water in order to break down a molecule, and we call that a hydrolysis. What we are talking about here is a phosphorylasis, which means that an inorganic phosphate is used in the reaction and not water. So the enzyme uses a cofactor called pyridoxal 5-phosphate, or PLP, to cleave glycogen phosphorylatically. So no water is used in the cleavage reaction, but an inorganic phosphate combines with the non-reducing end glucose to give glucose 1-phosphate. So the glycogen phosphorylase cleaves glucose from the non-reducing end of glycogen molecules. So if you have your glycogen chain over here, you have your non-reducing end residue. That is what gets removed by glycogen phosphorylase. So the amylase we spoke about was an endo-acting enzyme. It cuts somewhere within the chains of glycogen. This is an exo-acting enzyme. It actually needs a chain end, which is why as we showed earlier, all of those branch ends that the glycogen particle has, that the granule has, that is why it is important to have all of those chain ends, because the most important catabolic enzyme of that particle is the glycogen phosphorylase, which is an exo-acting enzyme. In other words, it needs chain ends. So it cleaves glucose from non-reducing ends of glycogen molecules, and it provides a metabolic advantage because the product is a sugar phosphate, which is a potential glycolysis substrate, and I'll say a little bit more about that now. What happens then is the enzyme phosphoglucomutase again then converts glucose 1-phosphate to glucose 6-phosphate, which is fed into glycolysis for energy production. So why do we call this a metabolic advantage? So you'll remember when we spoke about glycolysis, we said that the glucose is brought into the cell. The first step, catalyzed by hexokinase, phosphorylates glucose to glucose 6-phosphate, and that actually requires an ATP. Now think about what happens here. We have glycogen, and that is broken down by this enzyme called glycogen phosphorylase, and it causes it yields something called glucose 1-phosphate, which phosphoglucomutase then turns into glucose 6-phosphate. Now, but neither of these two processes, neither the glycogen phosphorylase nor the phosphoglucomutase, actually requires any input of energy. So it doesn't require that ATP to be spent. So whereas in glycolysis, you have glucose broken down to pyruvate, you invest 2 ATPs it yields 4 ATPs for a net gain of 2 ATPs. When you start with the glucose as part of a glycogen molecule, you use the glycogen phosphorylase enzyme and the phosphoglucomutase enzyme to turn glucose 1,6-phosphate into glucose 1-phosphate into glucose 6-phosphate. You haven't spent that ATP yet. In other words, it only requires one input of ATP, which is the phosphofructokinase, the third reaction of glycolysis, and you're still going to yield in the second part of glycolysis those 4 ATPs. So the net gain of ATPs in this breakdown reaction is then actually 3 ATPs, and not just 2 ATPs as it was with normal glycolysis starting with glucose. And so that is why we say there's actually a metabolic advantage in terms of energy gain when the substrate is glycogen and you use the glycogen phosphorylase enzyme to break down that glycogen. So the next important enzyme is, of course, the debranching enzyme that I mentioned earlier. So the phosphorylase and the amylase can't cleave past the branches. Those alpha 1,6 branches is problematic for both of those two enzymes. So the phosphorylase will stop at 4 residues away from the branch. So if this is our glycogen molecule, we have 2 branch points over here. We have our phosphorylase was acting from this side, but it will stop 4 residues away from the branch. And this, of course, what is left then is what we call those limit dextrins. Now, the debranching enzyme has basically got 2 activities. It's got a transferase activity and it's got a cleavage activity. So the transferase enzyme will move 3 of these residues to an adjacent branch, leaving just 1 residue. So of the 4 residues that basically scared off our phosphorylase, 3 residues are transferred to a different chain end. And all of these will now again be in the alpha 1,4, or they will all be alpha 1,4 bonds, which will allow the phosphorylase to work again. So in other words, there are 2 activity for the debranching enzyme. It transfers a trisaccharide group, and it also then cleaves away that remaining single glucose from the chain. So the single glucose that remained after it moved that trisaccharide is now cleaved off by the debranching enzyme, and it is released as glucose. So the debranching enzyme has a transferase activity and it has a glucosidase activity, and this is how it completely removes a branch and allows the completion of the degradation of glycogen. So we can then look at a brief summary of glycogen metabolism now that we have information both of how glycogen is made and how it is broken down. We start here with glucose 6-phosphate, and we will see that the phosphoglucomutase enzyme can either turn glucose 6-phosphate into glucose 1-phosphate, or of course it can turn glucose 1-phosphate into glucose 6-phosphate. And this is basically directed by what the energy needs of the cell is. If the cell already has a lot of carbon and has a lot of glucose 6-phosphate, it'll direct metabolism into the anabolic pathway for making glycogen. Although if the energy need of the cell is low, there won't be a lot of glucose 6-phosphate. There will be breakdown of glycogen. In other words, there will be more glucose 1-phosphate, and then the phosphoglucometers obviously acts in the reverse direction to make glucose 6-phosphate. Now, remember also when we spoke about gluconeogenesis in the previous lecture, we said that turning glucose 6-phosphate into glucose actually requires the glucose 6-phosphate to be carried into the ER lumen. There's an extra enzyme there that turns glucose 6-phosphate into glucose, and that allows the transfer, or then there's a transport system that allows the transfer of the glucose both out of the ER and out of the cell. But we don't actually need, in terms of making glycogen, we don't actually need glucose. The source for making glycogen can either be from glucose coming into the cell, being turned into glucose 6-phosphate, and then from there being turned into glucose 1-phosphate, or it can be from gluconeogenesis, which will act all the way up to glucose 6-phosphate, and then the carbon will be directed by a phosphoglucomutase to glucose 1-phosphate, and that will allow us a source of making glycogen. So when we have glucose 1-phosphate, the enzyme UDP glucose pyrophosphorylase will use a UTP, release a pyrophosphate, which is then eventually split into two inorganic phosphates. The glucose 1-phosphate and the UTP is turned into UDP glucose and the pyrophosphate. And of course, this is the building block for our glycogen anabolism. The enzyme glycogen synthase and the branching enzyme are then mostly responsible for producing glycogen, with that glycogenin primer, of course, that we spoke about. And as the glycogen chains grow, UDP is released. Once we have our glycogen and the cell is signaled that it is now in a low energy phase and it needs glycogen to be broken down, when glycogen is mobilized, it requires both the branching enzyme and glycogen phosphorylase, and of course amylase, although that plays a more minor role, to break down glycogen to glucose 1-phosphate again. From there, it can be turned by phosphoglucosamidase into glucose 6-phosphate, and from there it can carry on with glycolysis. Okay, so let's look at the mechanisms of how glycogen metabolism is regulated. Digestive breakdown of starch is unregulated, which means that nearly 100% of all starch ingested in our foods is absorbed and metabolized. But tissue glycogen is an important energy reservoir, and its breakdown is very carefully controlled. It is, as we have seen with other metabolic processes, under a reciprocal regulation through allosteric regulation and covalent modification. So again, there will be a local allosteric control of the enzymes and a global control through covalent modification, specifically through phosphorylation. So both enzymes are never fully active at the same time. In other words, the enzymes are under control when glycogen synthesis is switched on, glycogen degradation is of course switched off, and vice versa. So the allosteric regulation, it happens at the glycogen phosphorylase. We said the most important enzyme for breakdown of glycogen. The allosteric regulation of glycogen phosphorylase, ATP and glucose 6-phosphate are allosteric inhibitors. So think about that. It means that if there's a lot of ATP and a lot of glucose 6-phosphate already, that the cell doesn't actually need to break down its glycogen store. Okay, so it already has a lot of energy available in the form of ATP. It already has a lot of glucose 6-phosphate, which can continue its flux through glycolysis in the TCA cycle, etc. So it doesn't need to break down its glycogen store, and therefore it will inhibit the glycogen phosphorylase enzyme. However, conversely, AMP is an allosteric activator. AMP, of course, the main signal in the cell that the energy conditions in the cell is low, and therefore it'll have to switch on processes which will allow the cell to make more energy, and the breakdown of glycogen, of course, being one of those. So when ATP and glucose 6-phosphate are abundant, glycogen breakdown is inhibited because there's already enough energy. But when cellular energy reserves are low, glycogen catabolism is stimulated and glucose is released for glycolysis. There's also, as I said, covalent modification through phosphorylation of the enzymes. Glycogen phosphorylase has two forms. It has a phosphorylase B and a phosphorylase A form. In the B form, which is unphosphorylated, the enzyme is less active. So in the A form, the enzyme is phosphorylated and it is more active. So keep in mind that for the phosphorylase, phosphorylated means more active, and dephosphorylated or unphosphorylated means less active. And compare that now with the glycogen synthase, which is the regulation point of the synthesis. So we said, of course, we have this reciprocal control, we have regulation of the phosphorylase, and we have regulation of the synthase, and we see how these two play off each other. So glycogen synthase also exists in two distinct forms. It has an active dephosphorylated GSI form and a less active phosphorylated GSD form. So the synthase in its dephosphorylated form is active. The phosphorylase in its unphosphorylated form is less active. Okay, right, so we have that reciprocal control there. So the phosphorylated form is allosterically activated by glucose 6-phosphate, which means that, of course, glucose 6-phosphate is one of the points of, or one of the building blocks of glycogen. So when there's a high concentration of glucose 6-phosphate, it'll allosterically activate this phosphorylated form of glycogen synthase. Dephosphorylation is carried out by a phosphoprotein phosphatase called PP1, and PP1 inactivates glycogen phosphorylase and activates glycogen synthase. So how do those phosphorylation and dephosphorylation reactions happen? Well, as we've discussed previously, of course, this global type of control happens through hormonal regulation of glycogen synthesis, and we will discuss now the hormonal regulation of glycogen synthesis and degradation. So storage and use of tissue glycogen, maintenance of blood glucose concentration, and other aspects of metabolism are of course regulated by hormones. The most important hormones that we're going to talk about here is insulin, glucagon, epinephrine, and glucocorticoids. Now, in the previous lecture we discussed the role of glucagon in gluconeogenesis, and we will focus a little bit more on insulin here. So insulin is a response to increased blood glucose. It triggers glycogen synthesis when blood glucose rises, and between meals your blood glucose level should be between 70 and 90 milligram per deciliter. And when you have a meal, glucose levels can rise to 150 milligram per deciliter and should return to normal within 2 to 3 hours. What glucagon and epinephrine does, it does kind of the reverse of insulin, in that it stimulates glycogen breakdown. And that, of course, happens when blood glucose levels is low. But insulin, we've said, is the primary hormone responsible for conversion of glucose to glycogen. It is secreted by the beta cells in the pancreas. Secretion of insulin is a response to increased glucose in the blood. When blood glucose levels rises, insulin is secreted from the pancreas, and it lowers blood glucose concentrations in several ways. First, it does so by stimulating glycogen synthesis. Also, at the same time, because of that reciprocal control we just spoke about, inhibits glycogen breakdown. It induces synthesis of glycolytic enzymes, and this is kind of a summary of what happens. The metabolic effects of insulin are mediated through protein phosphorylation and second messenger modulation. We're not going to talk about those phosphorylation cascades, which happens between the insulin signal and the actual action on the different enzymes where they have an effect. But protein phosphorylation and second messenger modulation has these effects on the cell. First of all, it'll activate active transport. Now, what we mean by this is the cell will actually spend energy in the form of ATP to actively bring more glucose into the cell. So remember what insulin's job is. Insulin has to tell the cell to take care of this excess glucose that's in the blood. Insulin is a signal response to increased blood sugar, and it has to help clear this increased blood sugar. So it does everything that it can to help clear this high blood glucose level. So the first thing it does is it increases the active transport. So that'll allow the cell, even though there might already be quite a lot of glucose inside the cell, it'll tell the cell to spend ATP to bring more glucose into the cell. It'll also increase glycolysis, and at the same time also increase glycogen synthesis. So this kind of seems like it ends with each other, or at odds with each other, but it's actually not. It'll increase glycolysis, this degradative pathway, and because of course that gets rid of glucose. But glycogen synthesis, remember, also gets rid of glucose. So it'll use the glucose in the form of glucose 6-phosphate, which phosphoglucomutase will then turn into glucose 1-phosphate, and then we can start creating the building blocks for making glycogen. So both of these are actually ways of decreasing the glucose concentration in the cell, which of course the cell is now more actively bringing in. It'll also spend energy on making lipids, and it'll turn down lipid breakdown, it'll increase protein synthesis, and it'll decrease gluconeogenesis. Of course, it has to decrease gluconeogenesis because we're already in a condition where we have a lot of glucose. We want to get rid of any extra glucose, and we certainly don't want to turn on a process which is actually going to make more glucose. Okay, so those then are the effects of insulin. So just as a little bit of extra interest, for normal insulin glucose activity, a glucose tolerance test is applied to see if a person has become diabetic, or if a person has the condition of hypoglycemia. So how this is done is that a person is given an ingestion of glucose. You normally just drink a glucose solution, and then they follow the blood glucose levels that you have over a period of time. So when we plot the blood glucose levels over a period of time, from those measurements, a normal subject will show a rise after they've ingested the glucose, but it should return actually to normal blood glucose levels within about 3 hours. However, a diabetic subject, the blood glucose will rise higher and remain high for a very long time, and that is what we call hyperglycemia, which can be very damaging in various forms throughout various places in the body. Okay, so the next two hormones that we mentioned were glucagon and epinephrine. So in response to decreased blood glucose, we said that glucagon is released from the alpha cells in the pancreas. And one of the effects is that it will act in the liver and adipose tissue. Glycogen breakdown is stimulated, and also gluconeogenesis is stimulated, and this will allow glucose to be released into the blood. Remember, glucagon being a hormone that is produced when blood glucose is very low, and those effects will allow the cells to secrete glucose, and that will allow blood glucose levels to rise again. So when signals from the central nervous system causes the release of epinephrine, which is also called adrenaline, from the adrenal glands, a cascade is initiated that activates glycogen phosphorylase and inhibits glycogen synthase. So that's epinephrine, and epinephrine acts on the liver and in the muscles because of course those are the places where glycogen is stored, and so it wants to activate glycogen phosphorylase, which means that glycogen can be broken down very quickly. And the reason it does this, of course, is that it is the so-called fight or flight hormone. It makes a lot of potential energy available for the cells in a very short amount of time. So even though these hormones seem to do a very similar job, in other words, raising blood glucose, glucagon is involved in long-term maintenance of blood glucose, whereas epinephrine results in a very rapid breakdown of glycogen and a very short spurt of energy. Okay. Finally then, we look at the glucocorticoids. These are steroid hormones that control the expression of many genes, including many involved in stress and anti-inflammatory actions. Cortisol is a hormone that stimulates gluconeogenesis and increases glycogen synthesis. So we can ask the questions, why? What would the body try to do under high stress conditions? And why do you think stress leads to weight gain, as has been shown? Well, when cortisol is released under stress conditions, it will allow glycogen synthesis to take place, which means that because the body is under stress, it will tell over a long period of time, it'll basically ready itself for a later energy release. And the way that it does this is by switching on these processes that allows it to do both gluconeogenesis and glycogen synthesis, in other words, to store energy in this way. Eventually, when that goes on for a long enough period of time, it'll lead to weight gain, and this link between stress and weight gain has already been shown. Okay, so this is where I'm going to end this lecture. In our final lecture on this presentation, we will look at the pentose phosphate pathway and biosynthesis of NADPH.